if this piece here were aspirin,
Mhm.
then Humira, the best-selling antibody drug in the world, would be about the size of this entire building. The capital expenditure to build a facility is typically 60 or 70%, because you have to spread that over all the doses. You can get 10% of the cost out of the system
Sure.
by just continuous improvement. But if you want to take 2 orders of magnitude out, you can't just do the same thing in a more efficient way.
Right.
You have to wildly rethink things.
The shop down the way sells me a spirulina smoothie, but you're growing your drugs in spirulina.
That's it. Exactly the same stuff.
Cool.
Yeah.
All right.
Up until now, it's been so expensive to make biologic drugs that the only people who can afford them are ones with rich governments.
Mhm.
Our costs at scale are already well below $10 per gram equivalent. And it'd be nice if we can get the cost out of it to sell those at a price that is affordable, independent of government solvency.
Welcome to Development and Research, where we talk to people doing things differently in the world of clinical trials and drug development. My guest today is Brian Finrow, who runs a company trying to make drugs cheaper by growing them in giant vats of algae. I have that right?
That's right. Yeah.
Welcome, Brian.
Oh, thanks for having me.
So I want to start at the level of a single molecule, and we'll work our way up to the company and like a medical system.
Sure.
And with what might be a basic question, but what makes a molecule a drug?
Oh, well, that is, yeah, that is an ontological question.
Okay.
And it's much more complicated than you would think. Strictly speaking, a drug in the FDA rules and statutes is anything that is intended to treat or prevent or diagnose a disease.
But at this sort of structural level, when I like look down at the physical reality of something that we think of as a drug,
Yeah.
that's a molecule.
Yep. But then if you say, well, look at the molecule, what kinds of characteristics do the molecules have, regardless of subjective intent? We've basically got 3 kinds of drug in the world today. I'll put aside for the moment the first of these, the most famous, which are the vaccine molecules.
Sure.
The other 2, though, are small molecule drugs and biologic drugs, or large macromolecule drugs.
Mhm.
What differentiates the 2? Of course, from a regulator's perspective, they're very interested in the different characteristics, maybe personalities, if you will, that they have. But the main difference is just simply the size.
And the important part of the molecule is the shapes that the molecule has.
Right.
That's right. The penicillin molecule is just a weirdly shaped cluster of carbons, nitrogens, oxygens, whatever, that is made by a fungus in order to fight bacteria around it. Whereas when we're making a biologic drug, it's probably a protein. It's probably made by sticking amino acids, but these are big blocks. We're putting our structure together with like Legos, not by injection molding it into exactly the right shape. And now we have to deal with what the shape of a Lego is. And if we're building a big thing, I guess is fine. And
Exactly right. Yeah. So maybe just for a sense of scale, if this piece here were aspirin, then Humira, the best-selling antibody drug in the world, well, actually recently overtaken by Keytruda, but same size, would be about the size of this entire building.
Sure.
Yeah. So much, much larger, just entirely on a different scale. It also has important implications for manufacturing. So small molecule drugs, because like as you point out, you can, in most cases, synthesize them with chemistry, just like basic, like high school chemistry stuff, test tubes, mixing things together, presence or absence of a catalyst, heat them up, cool them down, different things happen. Those processes tend to be extremely scalable. And that's why, drugs like aspirin are very, very cheap, particularly after they go generic, and widely available. So aspirin's a good example. Antibiotics are another good example. Now, biologic drugs, by contrast, are so complicated that we can't use those kind of building block type chemistry approaches. The only way to manufacture those is in some kind of a living cell. And we have to hijack the existing machinery that nature gave us through genetic engineering to coax, in the case of Humira, it's a mammalian cell called a Chinese Hamster Ovary cell, into doing something you wouldn't normally do, which is to pump out a whole bunch of this monoclonal antibody protein that is the Humira drug. What makes antibodies great for this is they have some biochemical properties that we call affinity, avidity, which just means they're how much they stick, how hard they stick. But they also have this property of a specificity, which means they only interact with the targets they're directed at, by and large. And their antibodies are famous for this property, the specificity property. And what it means is that they're extremely unlikely to interact with anything else in your body and cause off-target effects.
It's not going to get stuck to the surface protein of one of my cells that needs to pump something to do something.
Exactly, yeah. So that means that as a drug, it decreases the amount of side effects and risks that you would expect to encounter in clinical use.
Yeah.
So antibodies are great for this application.
They also get treated differently by the liver. They get metabolized differently. There's beneficial effects there too.
If you inject them, antibodies are just proteins, and all proteins are just made of amino acids, like the rest of your body.
Right.
And so when they break down, they just break down into amino acids, which are nutrients.
And not like toxic half molecules that can be in my liver. And so we have this very specific sort of cell culture that someone captured, made tweaks. I mean, this is, a marvel of modern engineering, getting it to do this unnatural thing in that it's pumping out this antibody out of these Chinese hamster ovary cells. From where I sit, it's not clear why this is, I don't know, hard. So like a story that I think of is golden rice is genetically modified rice. It's rice that is genetically modified to produce vitamin A, so much vitamin A that it changes the color from white to gold. And this, has the potential to save countries from vitamin A deficiencies across their population with no one having to do anything in a factory. Like these Chinese hamster ovary cells are grown in very, very controlled conditions. But if you want to deliver vitamin A, you can just pack it into rice and then grow it by the acre. So like, where's the breakdown here from like, if I want to deliver more vitamin A, I can just put it in the rice, put the rice in the field. So like how it is that we need to treat Chinese hamster ovary cells to make Humira antibodies.
Well, I can answer that coming at it from a couple of angles. Sure. The one is, one doesn't simply genetically engineer rice. It's actually quite difficult to engineer.
It's another marvel of the modern age.
Exactly. Even in the era of CRISPR, this isn't a field that my company does work in, but somewhat adjacent. My understanding, the most recent report I read estimated it's probably still $100 million to build a stable, scalable, genetically engineered strain of something like rice.
Okay.
It's quite difficult. Now, what makes it very scalable to manufacture once you have done that is the fact that, well, we've got maybe 8 or 10,000 years of human ingenuity into making that a scalable system.
Right.
In terms of domesticating rice. Now, we haven't done that for 10,000 years with Chinese hamster ovary cells.
Maybe we will be better at it, 10,000 years from now. Whereas now that like the growing conditions here are like very intensive laboratory conditions. You set the sort of scene for that. Paint us a picture.
Yeah, it's the opposite of planting a bunch of rice out in a field, right? It's these hamster ovary cells. I mean, really, if they had their druthers, I think they'd prefer to grow inside a Chinese hamster's ovary.
Sure. Yeah.
Right? So instead, we have to trick them into growing in a giant steel tank, and that's not so easy. In addition, the hamster ovary cells, they're evolved in that particular setting to make the proteins that a Chinese hamster ovary needs, not human monoclonal antibody. So we also have to trick it into that second trick. All of that requires an immense amount of effort. And the tricks involved really explain why it is that over 100 years elapsed between when, in the late 1800s, when Paul Ehrlich first coined the phrase magic bullets to describe antibody drugs.
Describe antibodies.
And the 1980s, when they got the first antibody drug into human clinical trials. So that's all part of the manufacturing side. But on the other end of the spectrum, then there's also the domain in which it's being used. So rice, golden rice, with this rice with vitamin A in it. Well, you're going to eat that, aren't you? People are going to eat that. And eating a protein, even a genetically engineered rice plant with genetically engineered parts to it, is, well, the default setting on that is safe. We eat stuff every day. And there's all kinds of contaminants in all of our food.
Many things with dirt in it, or they'll swallow a bug, it's fine.
And this is our bodies have evolved, to our digestive tracts evolved to handle that. Now, if you're going to go into this other domain, which is we're going to inject this foreign bodies, essentially, which is what drugs are when you inject them, then again, you have to be extremely careful because we know a lot of ways to hurt people
Mhm.
by injecting the wrong thing accidentally. So, for just one concrete example, there are mammalian viruses that will infect Chinese hamster cells and also human cells.
Mhm.
Got to be extremely careful that those don't get into the vial that gets injected.
Especially if you're injecting them straight,
Yep.
where eating them, you'd have some lines of defense,
Right.
like acid and
Yep.
immune system.
Exactly.
Sure.
There's also, you can get, ground glass. You can have accidental mix-ups with the proteins are not just dry powders, right? They're mixed into a solution.
There are a whole bunch of
There's
other things,
certain ways.
other proteins and lipids and whatever that the ovary cells.
Oh, you got to be very careful to separate that out. Yeah, particularly insulin isn't made in Chinese hamster ovary cells. That's made in E. coli. E. coli manufactures this other molecule called LPS, lipopolysaccharides. These things are extremely immunogenic if you inject them.
Mhm.
People get very sick if you're not careful about getting it all out.
Mhm.
So yeah, it's both the difficulty of making it and the difficulty of, once you've made it, making it safe to inject. And all of that adds up to cost and difficulty and challenges in the drug development process as well as the drug manufacturing process.
So this brings us to talking about Lumen. This things that talking about your company, Lumen Bio. That's Lumen, not Lumon, for
Right.
all of our Severance watchers out there. But Lumen has a particular approach to this, where you are doing something that is more like growing drugs in golden rice,
Right. Yeah.
than in steel vats that are, where you're applying tricks upon tricks.
Yeah. So what we do is manufacture our biologic drugs. So they're similar to maybe Amgen's with their Chinese hamster ovary cells, in that these are biologics, right? So they're the big, kind of building size molecules, not the sculpture size molecules.
Mhm.
But a key difference,
And they're being built by living things.
That's right. They're also being built by living things. But the two key differences are, number one, we're not making these for injection. These are all of our programs currently in the clinic are for disease targets that are in the GI tract or GI tissues. So oral delivery. And second big distinction is, rather than using Chinese hamster ovary cells or E. coli cells, these things, the workhorses of the biotechnology industry, we work with a different microbe called spirulina, which is itself a food.
The shop down the way sells me a spirulina smoothie, or it's a kale and spinach and spirulina and whatever, green apple. It's all these things. But you're growing your drugs in spirulina, which people put in my smoothie as a
Yeah.
health food additive. What's it?
That's it. Exactly the same stuff.
Cool.
Yeah.
All right. Why spirulina? Why spirulina? Why not rice? I think we understand why not Chinese hamster ovary cells. But you want something that grows. Why spirulina specifically?
But people have tried this idea in rice. Actually, rice is a good example. There's a Japanese pharma company called Astellas that does a lot of work actually trying to make different therapeutic proteins, even vaccines in rice seeds. People have done this with tobacco leaves, actually, with corn, potatoes. So there's some very special things about spirulina that make it particularly useful for this kind of application. Just to begin with, well, if you're growing your things in, say, rice, let's stick with that for example. If you're growing it in the rice seed, well, this is a little bit of a shame because you're going to grow a lot of rice material that you're not going to use.
Sure.
Most of a rice plant is not, in fact, rice. And so you're going to have to grow whole fields full of it and cut it down and throw most of it away. So there's a kind of an efficiency element there. Spirulina, by contrast, is a single-celled organism. An algae, green algae, salt water.
Sure.
Very simple. It's got another advantage, which is a little bit more technical. If you think about what's in most foods, potatoes or rice, whatever, it's either almost always carbohydrate. And less common, like soybeans, there's a lot of lipids in there. And that's because carbohydrates and lipids are a good way for plants to store energy for the long term.
And in terms of humans cultivating the plants, it's good for the things that we want the plants to have.
So with spirulina, by contrast, is about 70% protein.
Okay.
And it's very
Which is why people keep trying to put it in my smoothie.
exactly. Yeah. It's one of the, well, there's also a lot of other vitamins and things in there, but the protein is what's useful for us because remember the drugs we're making are proteins. And so if you think about these things, it's just miniature little protein making machines. They just love cranking out lots of protein.
Whereas there's a lot of protein. So if I hijack a quarter of the protein, I'm going to get a lot of protein. If I hijack a quarter of the protein in a, rice plant, I'm going to get a tiny bit of protein.
And this really matters because both in terms of cost, of course, because the more protein you get to express in your cell or the, whatever your host organism, then the cheaper it is. You're not wasting resources growing other parts of the plant that don't have utility. But it's also important because remember, we need to get this into a capsule. And so with spirulina, our best performing lab streams, we've gotten as high as 25% of dry biomass as soluble bioactive protein. The highest I think I've ever seen reported in a rice stat was maybe 0.1%. So dramatically lower. And
you haven't improved it by 25 times. You've improved it by 250 times.
Yeah.
Okay.
Yeah.
Great.
Yeah. It's just much better at this particular task.
Yes.
Than rice. So even though rice is cheaper, I mean, you can buy rice cheaper than spirulina in the grocery store for this application. That's a big advantage. Now, what you're buying there, this almost certainly comes out of the Imperial Valley in California.
Okay.
They have this huge outdoor ponds of 100 acres or so of them.
Sure.
Yeah. And they grow it there. So which is another clue. Spirulina is the only microbe that is farmed at such a scale. Any other microbes are using, in fact, all of the three workhorses in biopharma, the E. coli and yeast and Chinese hamster ovary cells, they have to do everything in sterile tanks. But think about it this way. So that they can make that cheap enough to sell in the grocery store.
Right.
Which is a clue.
Right.
To the scale they must be growing it at because, economies of scale are the flip side of scale.
And they're either filtering out the E. coli that makes it into the pond or the E. coli never makes it into the pond. Well, they're big open air pond. The E. coli is going to drift in. So
there's all kinds of stuff in there. In fact, this particular one is on a migratory bird route.
Nice. There's some particularly biological things.
It comes out of it in the summer.
Nice.
Yeah. So no, all kinds of stuff in there.
But this can just go through filtration and at the end they end up with a product.
Spray dry it. But you don't want to think too much about what's on your food. It's not extremely clean. It's just actually a testament to how well evolved our GI tracts are.
Sure.
So we're not all sick constantly.
Right.
So this is a clue, though.
So your spirulina vats are cleaner than the big open air ponds on the.
This is the point I want to emphasize is that we're not growing in the pond with birds.
Sure. Inside,
it is, I think, remarkable that even with that production system, it is totally safe.
Right.
Now, of course, we're making biopharmaceuticals. Some of these go into people that are very sick.
Right.
So they're very vulnerable. Obviously, we grow indoors. Or we control all of the inputs in the environment. But the key thing is it's not necessary for it to be sterile in our system. It's indoors much cleaner, but sterility isn't required. And that's a key to getting the cost down and the scale up.
So rather than stainless steel vessel, it's got all its, and then we have to sanitize it afterwards and maybe we failed and a little microbe comes in. Instead, your spirulina vats, this is technology that beer brewers or whatever are using.
Our equipment is even cheaper than what
than brewing beer.
Yeah.
Excellent.
Yeah. Yeah. No, the capital expenditures, the cost of buying all of that gleaming stainless steel and all the piping and things, particularly on the downstream side. If you break down the cost of one dose out of a biomanufacturing process, the capital expenditure to build the facility is typically 60 or 70%. Because you have to spread that over all the doses they make over the lifetime of the factory. That's how the accounting rules work. It's typically 60 to 70% of it is the equipment and the capex. And so if you want to get, as our goal was to get the cost, per pure gram of protein therapeutic equivalent down by at least 100-fold.
Sure.
Then you cannot do that unless the capital expenditures are dramatically lower than on a traditional system.
Right. So I've been to your factory floor. You sort of took me on a tour there. And you're set up in a former bakery in Seattle.
Right, yeah.
Pre-revenue company. And so you have not sunk all of that cost in. But this is because of the organism that you're choosing to grow it in.
That's why
just been enabling all of that and making it possible.
Yep.
Why isn't everyone doing this?
Everyone tried in the field. Yeah. So we have some very nice IP around the technology necessary to bioengineer the cells to make a therapeutic drug protein. My co-founder, Jim Roberts, and one of my former colleague, Ryo Takeuchi, they're the two inventors on the patent. It was a real breakthrough. You see, people have known, like spirulina has been on the market in the US since the 70s.
Sure. And people have eaten it for centuries. They farmed it and eaten it for centuries.
And people have been making, since the 1970s, right? They've been making recombinant protein therapeutics too, right? And so this is kind of an obvious idea. Why don't we use spirulina to make, put the two together? And the challenge, though, many very well-funded groups tried and failed over the years. And occasionally they do us a favor. They publish their failed attempts, which scientists usually don't do, which tells you something about how often people came back to this problem. And none of them succeeded, for half a century until Jim and Ryo, they figured it out. So in a way, the company we aim to emulate is Amgen. And like Amgen, we are built on a fundamental breakthrough in the cell engineering technology. For Amgen at the time, the new technology on the scene, new cell engineering technology was mammalian cell engineering. And that's what allowed them to make EPO and then monoclonal antibody drugs, Enbrel, and all these other things. It was a real breakthrough that set the stage for sequential innovation by opening up a new field of innovation. Very similar, actually, to Genentech also, which was just founded on the late 1970s, then new technology for the first time engineering E. coli. So that's a grand tradition we aspire to carry on.
Where the story is, we can manufacture something that we couldn't before. We can manufacture something in a way that we couldn't before. And now, with that power, there are any number of drugs that can come after that and address things that couldn't be done before. It's like those drugs, it was not possible to produce them at all, at the right scale, at the right cost. Those drugs are made possible by this breakthrough in manufacturing.
Yeah, that's exactly right. Yeah. But to understand why, so we're not trying to put these other platforms out of business.
Sure.
So so I'll start with E. coli. E. coli that came out in the 1970s. The very obvious thing is to make insulin.
Sure.
Before then, the world's got its insulin from pig pancreases.
Like you have to grow a whole pig,
and
then you slaughter the pig and grind it with the pancreas, and you get my insulin.
Squeeze out the insulin. Anyway, it's much better insulin today.
Because we have to grow the E. coli and not
just grow the E. coli, and you don't have to waste all the time going up the snout and everything.
Yeah.
So we had, the reigning drug technology at the time was small molecule drugs. That's all the antibiotics and things. These guys were all ruling the roost. The important insight here is that Genentech, it's not like they shut down or outcompeted the legacy technology. In fact, we're still making small molecule drugs, and there's still new drugs coming out on those technologies. It's just that you want the new technology. E. coli allows you to do a thing that you couldn't do with the.
So it's just adding to the field. We can do something. It's maybe putting the pig insulin industry out of business, but
it did put the pig insulin people out of business. Yeah, that's true.
But it meant that so many more people could be reached by insulin. So many more patients could be helped by that.
Eli Lilly is who got that IP from Genentech, and they're a $700 billion company now. So they're doing fine.
Yep. Yeah. They've done a few things after that recently, especially recently, but yes.
So then in the 80s, then they came up with mammalian cell engineering. And again, there's things you can do in mammalian cells you can't do in E. coli. Make a monoclonal antibody. So it's not like Amgen put Genentech out of business.
Right.
There's just a whole new wave of innovation that came out of that. And interestingly, it wasn't until maybe 10 years ago that for the first time, a majority of new drugs approved by the FDA were actually biologic drugs.
Okay. But we have gotten to that point where the majority are made out of these big things made out of biological building blocks built in biological processes.
Yeah. So the E. coli people are still in business. The small molecule drug people are still in business. It's just we're making the pie bigger and bigger. And so it's in like fashion now. So what's the killer app for us? Well, so we'll start with the easy things. So our most advanced program is directed at a disease called C. difficile infection.
Okay.
So C. difficile infection is a bacterial infection of the GI tract.
Okay.
And so it's great for us.
If I get a bad bacteria in me, then it starts to colonize and grows, or who gets it?
So C. difficile is a bit interesting because about 10% of people are at any given time have a little bit in them.
Okay.
It's all around us in the environment. You can't avoid it.
Sure.
And particularly if you visit hospitals.
Okay.
So normally it's fine. If you have a healthy immune system, if you're sort of a in mid-30s, healthy adult with a good Western diet, you're fine. It doesn't trouble you at all.
And it's just in this ecosystem of bacteria that are in my gut, and they're in balance, and everything is
typically
good.
happens is that it's typically an older person. They have weaker immune systems. They go to the hospital. They get antibiotics, usually for something unrelated,
Sure.
hip injury or a hip surgery or whatever. And
But they get oral or systemic antibiotic.
clindamycin in particular is notorious for this. So they get their clindamycin. What happens is it clears up their infection, say their post-operative surgery wound, but also kills off your GI microbiome. And it turns out that's very important for keeping this C. difficile, which is all around us and constantly being exposed to it. That's what keeps it at bay, is our commensal bacteria crowded out. And so when your commensal bacteria damaged by antibiotics, then this C. diff kind of moves in, sets up housekeeping, starts pumping out this toxin, TCDB it's called. And that toxin breaks down the cells lining your gut, and then you get all these terrible symptoms, diarrhea, culminates ultimately in, well, a lot of fatalities, but also colonic resection surgery, and you live with a colostomy bag the rest of your life. So it's really terrible.
Yes.
Okay, so that's the disease. What's it got to do with us?
Right.
Well, remember what we're doing here is we're using this food organism that we can now engineer and grow safely with these drug proteins. And in this case, we're making these proteins called monoclonal antibodies. They're not strictly antibodies, kind of like it. And what these do is just like during COVID. Remember we had the antibodies against COVID spike?
Yes.
Everybody learned about that. So the antibody binds to the COVID spike. It can't infect your cell. Antibodies are great at this. Antibodies are what your body uses, manufactures anyway,
Sure.
to fight off viral or bacterial infection.
By sticking to them. This is what they're engineered for.
Yeah. So what we do is we find an antibody protein that sticks to the part of, in this case, it's the toxin we're neutralizing, and it sticks to the part that causes the gut lining cells to break down, and it neutralizes them. So it's like an antidote to the toxin.
They pass through the stomach and the acid and all of that.
This is a funny thing, a very serendipitous discovery. We didn't know this going into our first clinical trials, but actually, the way we make these, the protein, the drug protein molecules are actually inside the cell.
Right.
And then they're dried. And so it kills the cells. The cells aren't they're not crawling around inside you or anything like that. They're dead cells. They're inert. But nevertheless, they're intact. And so it's like a little bioencapsulation. And it turns out in the low pH of the stomach,
The acidity.
exactly. The acidity of the stomach, it all stays kind of together until it's released into the small intestine, which is where most GI infections happen. And there's a big change in the acidity. It gets much more neutral, and then the cell breaks open. It's very elegant, actually. I wish I could say we designed it that way, but it's a convenient coincidence.
Yeah.
And
This is just how my body is breaking down all different kinds of the foods that I eat, or the plant cells, or the
exactly.
Great. But did this particular one, or something about the nature, the character of the spirulina cell, is it's mostly getting released in the GI tract. And so if you want to go into the GI tract to fight an infection with the proteins inside, you're golden.
It's perfect.
Fantastic.
So and we've published all of this data. So it's really kind of an elegant solution, sort of a one-stop shopping. Makes it very inexpensive for us to make these products.
Right.
Yeah. What makes it particularly well suited. So now coming back to your original question, is what do you do that the other older platforms don't do well? This is something we think that's just dead obvious. Well, think about it. This is a disease of the GI tract. This bacteria are in the tube, the lumen of the GI tract, and so we can put the antibody drugs right there, where they are.
Right.
Now, contrast that with if you injected antibody against C. difficile infection.
Right. It goes in here.
It goes in over in the arm, usually. And almost all of that antibody protein stays in the blood.
Right. This is going to diffuse through like 8 liters of blood. And then some of it's going to be in my gut, I guess.
Very small amounts.
It's going to be on the outside of my gut, not on the inside of the tube.
Yeah. Scientists have a term for this. They call it the partition coefficient.
Okay.
It's, for any given protein, what is the percentage of the concentration of the drug in the blood serum that you inject, and what percentage of that concentration do you achieve in what's called the interstitial tissues.
Mhm.
And it's something like, estimates vary, but between 1% and 1/10 of 1%.
Wow.
And remember, too, for C. diff, the disease isn't in the interstitial tissues. That's the
Right.
bit between the blood vessel and the GI tract. It's in the
It's on the
tube.
outside of my body.
It's on the-
Right.
Yeah.
The inside of the tube is
Yeah.
topologically the outside of my body.
So that's an interesting way of putting it. Yeah, you have to inject the drug and hope enough of it leaks out of your body
Right.
to get to where the infection is.
Yeah. And so you would be lucky if 1% of it leaked. And so you need to be treating it at 100 times dose here.
So
Right.
this is an intuitive way to understand, and particularly for C. diff, because actually a large pharmaceutical company actually did design and develop monoclonal antibody, IV-infused monoclonal antibody drug for C. difficile infection.
Systemically injected.
And they IV infused it, and it's just exactly what you said. You got to inject enough of it. So the dose size there was 15 milligrams per kilogram, 15 mg per kg, which is a whole gram of pure antibody protein was the dose.
Mhm.
And this is actually the very few monoclonal antibodies that are approved for infectious diseases. And this is actually something they have in common, these huge, huge doses. The top dose
Sure.
in Regeneron's COVID trial, which has the same issue,
Mhm.
right? Airways.
Oh, right. Yeah, I guess.
Epithelium.
Yeah, I'm injecting it and I hope it leaks out of my, or to the inside of my lungs. Okay.
So the top dose in their phase 2 was 15 grams.
15. Oh, so that's 15 times.
15 grams of pure antibody protein.
Okay.
Yeah, huge, just
Okay.
huge. And so we actually have, because this large pharma company put so much work into this, we have actually some really nice data. And they measured, they looked in the stool of these patients with C. diff, and they were only able to detect very minimal quantities in fewer than 10% of the subjects in the study.
And 90% of them, they couldn't find it.
They couldn't see any out of the stool pit, and the amounts there were not even large enough to quantify. So just minimal trace amounts. So it's kind of remarkable that that drug worked actually.
Yeah.
Now compare that with what we do, and we can just fill up the whole tube.
Yeah.
Yeah.
And maybe 1% of it leaks in the other direction, I guess.
Yeah, no evidence of that, but
Okay.
surely it could be happening.
Or in 10% of patients,
Yeah,
unquantifiable, tiny amount maybe
Yeah.
leaks in the other direction.
it could be. But in general, we get these two pretty nifty advantages for a disease like this, which is that we get a better shot at efficacy
Mhm.
because simply pretty much every drug has an effect that is dependent on its concentration at the
Sure.
site of disease. So we can get more concentration where it's needed.
But there's some limit to the concentration you can give either by the economics of manufacturing or by the side effects rising at the same time.
That's usually that's the second thing, which is the side effects are typically driven by exposure of other tissues in your body.
Sure.
And by keeping it in the GI tract, you see, we avoid those exposures.
Okay.
And we cut down on the risk of off-target effects and off-target toxicities. So in short, particularly for a GI disease like C. diff, it's a way of making a drug that is everything you want. It is more likely to be more effective, less likely to cause off-target toxicities and side effects and things. And on our platform, because of what we were talking about earlier with the CapEx advantages and the scalability of it, remember, cheap enough to buy in the grocery store,
Yep.
It's also less costly to manufacture. And as a final thing, it's much more convenient to take oral capsules than to
Than an injection.
take an IV infusion
Yeah.
of a very expensively manufactured drug that circulates everywhere and doesn't mostly get to where it's needed.
Let's talk about how that shows up in the clinical trials that you run, because your clinical trial for treating C. difficile here is going to be different than someone who's running an injected antibody or something else. But even within that, there are design parameters. There are smart choices you can make and, well, less smart choices you can make. You can follow convention. You can do some things differently. What have you been doing differently in your clinical trials based on having all these advantages in order to play into them, or just because everyone else gets it wrong and you might as well get it right?
I should caveat this by pointing out I'm not a clinician.
Sure.
I trained as a lawyer.
Yeah.
And so I have a tremendous.
We'll talk about that in a minute. I trained as a quantitative trader. So, this is neither of us is clinician talking about the clinical trials here.
So in the case of this particular program, we're doing something that is, I think, quite novel for the industry. In that the drug product, which I've kind of lied a little bit. I've oversimplified exactly what it is. There's actually 4 proteins in it.
Okay.
There are, and this is something very different than what the industry does. 3 of them are these antibody-like proteins, and then they bind and neutralize toxin B, TCD-B.
Mhm.
This is what mediates pathogenesis in humans that are infected with C. difficile infection. And the fourth, though, is a different thing. It's an enzyme, another kind of protein. Enzymes are proteins.
Mhm.
And this enzyme binds to and interferes with the cell wall of the bacterium itself.
So this is what we think of as like an antibiotic mechanism.
Kind of, yeah. Yeah, but antibiotics are, I mean, that term is strictly speaking of small molecules.
Small molecules, and it's small molecules that are disrupting the.
It's an
Okay, it's an enzyme.
enzyme protein that has the important thing is it has a different mechanism
Mhm.
than binding the toxin. We didn't do this because we just want elaborate complexity in our
Or
lives.
yeah,
We think that this combination will be much more potent than just one or the other.
sure. I mean, this makes sense. I think my question when you say like, yeah, we just stick to the toxin. It's like, well, the toxin is just going to stick around. I'm going to leave all the bacteria there. It's going to keep pumping. Like, what's the end game here? But the end game is an enzyme to go after the bacteria itself, and hopefully my immune system is regrowing and my ecosystem is regrowing
But
eventually.
here we were encouraged. Remember, even in the big trial I was telling you about by this other company,
Sure.
non mostly non-detectable levels of this one antibody
Sure.
was enough to improve disease outcomes.
Okay.
So we are optimistic. We've got
Mhm.
3, and the 3 together are about 1,000-fold more potent.
Hmm.
They're measured, neutralizing power. So we are like pretty optimistic that we got something good going on.
What's the thinking on 3 there? So when people go after COVID with antibodies, is
Yeah.
it also 3? Is it one antibody?
Generally 1, sometimes 2. Regeneron made one with 2.
Okay.
Yeah.
Okay.
So what we're doing there that is quite different, though, is this combinatorial exercise.
Is this because there are 3 different types of toxin, or they need to work together?
Much more interesting than that, actually. But there's a little bit of diversity,
Mhm.
but that's not actually the main reason. What we've discovered is that, and we published on this as well. We have a preprint out there. This is working through peer review. Is that if you design an antibody cocktail like that, that's the FDA terms, a cocktail of antibodies.
Mhm.
What we found is that the artful combination like this can get us a measured potency that is thousands of fold more potent than any single antibody could ever achieve.
Yeah, like what is the thing that's going on there?
It's super interesting. It has to do with the behavior at the molecular level of an antibody binding event.
Mhm.
So when antibodies are binding, they're not like it's not like sticky tape. They don't just like bind and they're
And stay.
gone. That's a covalent bond. That's not what's happening.
Mhm.
These are non-covalent binding events. And when there's all kinds of parameters you can
Velcro. It could stick, but it could get pulled off.
yeah, they're kind of more like static electricity.
Okay. Okay.
Like it sticks
Like I stick the paper onto the ceiling, and maybe it'll.
it sticks for a while, but
Yeah. Yeah.
with antibodies, everything moves very fast at the molecular level. But the antibodies are basically they're coming on and off of their substrate all the time.
Mhm.
And you can measure this.
Mhm.
There are different tools analytically to measure the on rate and the rate at which they come off after the binding.
Mhm.
And all of this gets reduced down into a single figure that's most commonly reported, which is the affinity, or in the case of a multivalent antibody, the avidity.
Okay.
So this dynamic behavior is what's important to understanding why it is we can achieve such high potencies with what we call the synthetic avidity effect. Basically, there's 3, again, not antibodies, but antibody-like proteins. They're binding, if this is the toxin, they're binding in different spots,
Right.
and they're kind of coming on and coming off all at, you know, all constantly.
But this one is stuck on 80% of the time, and this one is stuck on 80% of the time. And
Remember, each of these alone has neutralizing potential.
got it. Okay.
And so what you want to maximize is the time that something is neutralizing it. But
Is this thinking about it? If it's like this one is has a 90% on rate, and this one has a 90% on rate, and that one has a 90% on rate, one of them is going to be stuck on 99.9% of the time.
Yeah. Yep.
And so I guess I could spend forever and a day trying to engineer an antibody which was so sticky that it stuck on 99.9% of the time,
Mhm.
or I take 3 of these on 3 different binding sites, which are each 90%, and I
Yeah.
get a 99.9% cocktail.
That's it.
Okay.
So at the end of the day, antibodies potency is measured in concentration terms. Yeah, how much drug dose of the cocktail do we need to cause the 50% maximal inhibition of cell intoxication of a certain amount of toxin
To make
TCG.
this amount of toxin half as deadly
Yep. Yep.
as it was.
Yeah. So this it's a little bit arcane, like I say, but to kind of boil it back up, antibody potencies are measured in concentration, so that the molar terms, in terms of the concentration of antibody for whatever.
This is how many antibody molecules I need per like
Yeah.
milliliter times some like
Yeah.
ridiculous number.
And so then a typical antibody you get out of like a machine learning or AI system will be a micromolar, low micromolar binder. A good antibody, a clinical grade antibody, will actually be nanomolar, 1,000 times better. A great antibody will be picomolar.
And that's 1,000 times again.
And there are very few examples of that.
Sure. Are the best selling antibodies
The antibody
way down there, or
the best selling antibodies are mostly in the nanomolar range.
okay.
Yeah. The antibody that I was saying earlier, the IV infused one that this big pharma was selling, actually just got pulled from the market. I think that was in the low nanomolar range. So picomolar is great. The measured avidity of this triple cocktail with this artful combination is in the femtomolar
1,000 again.
range. Yeah.
Wow. You're getting your effects at 1,000,000 times lower dose, at literally 1,000,000 times lower dose than where antibodies normally are on the market by doing this combination effect.
Yeah.
Sure.
So it's going back to to your original question, which is what's different about how you do clinical development? Well, this is something very different, and it's troublesome because each of these is in a different strain, which you have to grow more GMP batches,
Mhm.
blend them together. That's a lot of complexity.
Mhm.
But this kind of potency advantage, we think, is going to be important to the ultimate clinical efficacy.
Mhm.
Because remember, in the real world, I mean, this is all measured in a petri dish in the lab, right? In the real world, there's all kinds of other things going on,
Right.
including, for example, people don't always take their drugs.
Sure.
C. difficile infection in particular, it's predominantly an older crowd, and so there's a lot of pill burden among these individuals to begin with.
But they miss a day and
At times they're forgetful.
sure.
So adherence is what all of this, do people adhere to the dosing regimen?
Mhm.
And also it's a very messy environment. C. difficile is characterized by an awful lot of GI distress
Mhm.
and diarrhea and things. And so it's a chaotic environment that we're going into. And so we wanted to whack this disease as hard as we can.
Yeah.
And so that's why we have this complexity. The reason I think very few other companies do this. There's a couple of
Which was going to be my next question.
Yeah. Yeah. It's I think we're pioneers in this regard. It's very expensive to develop a single monoclonal antibody on one of these conventional systems.
Sure.
So we
And if you're proposing to do it 3 times, then
then it's 3
it
times
just
as
breaks
much.
the bank.
And because you're injecting them, people still encounter unexpected anaphylaxis, for example, with injected proteins.
Mhm.
That happened with a COVID program recently.
Mhm.
So if you inject 3 times as many proteins because you want to get the cocktail advantage,
Mhm.
then you've got a real risk that in the clinical trials,
Any one of them has a rare side effect and
then
you're dead.
stuck.
Yeah.
You're toast. Yeah. So it's a function of
whereas in your setting, you're just doing a much safer thing. And yes, it might be the case that one of the 3 of them has a whatever, but they were all very safe because you were putting them on the outside of your body anyway.
exactly.
Sure.
Yeah. So it's a big advantage.
So there are other companies that are running C. difficile trials that have
Mm.
candidates for whatever, and some of them are trying to treat straight to the GI tract. Are you fighting with them for patients? Like at the level of your trials, cancer trials are like infamously fighting each other for their patients. Are you in the same fight with the other companies developing C. difficile drugs?
Not really. There's only so many oncology centers in the U.S. and
Sure.
And so what I've heard, and we haven't run into this ourselves, but I've heard is that sometimes you'll find a situation where there's like 3 or 4 trials going on, for a single type of cancer, and
Right.
the principal investigators just have to like make hard choices about who goes into which trial.
Yeah.
So we don't have that issue.
Because C. diff is common in the specroxes, but also
It's just awfully prevalent. So there are a couple of other C. difficile trials recruiting right now, but none at the same centers where we are. And there's another distinction. There's been so remember what I was saying earlier about C. difficile infection, how it's this the cause of it generally is these antibiotics kill your microbiota.
Yeah.
Well, there's some other companies are going a different way, rather than the way we're doing, which is going after the bacterium and its toxin. What they're doing is they're saying, well, let's just fix what's broken. We'll accelerate the re-engraftment of the microbiome,
Okay.
and this is called fecal microbiota transplant.
Okay.
So you can get there in 2 ways: from the bottom up, right, or from the top down.
Neither one of those sound very pleasant.
But they put in a capsule.
Okay. I guess this makes the top down more pleasant.
And the jokers call it of course, a capsule,
Of course.
but it's fine. It's just a capsule.
Sure.
And
It's sealed all the way until it makes it into my gut anyway.
the insight is simply, okay, we've nuked your microbiome. Let's give you some starter seed.
Let's put some other bacteria there. They're the good bacteria that
Yeah.
live in human guts and fine.
Yeah.
Sure. Okay.
So the challenge, one of the challenges,
So that's a way to get to a good place, but it seems kind of indirect and fussy, and there's still toxin while you're dealing with the thing. And what if it doesn't take? And so,
yeah, and I think there's a practical challenge that we don't have, which is that of course you can't give antibiotics with it's fundamentally a probiotic. Think of the antibiotic.
oh, to be fair, I'm trying to like drop these bacteria and hope they survive, but I still need to be taking my antibiotic. I'm just
Right.
hosed
So
because they're not going to stick and survive.
exactly. Yeah. So it's a bit troublesome for the patient. They have to come back in. Oftentimes they have to do like a bowel cleanse to kind of wash out the antibiotics, or and do an enema application sometimes. But the bigger, more fundamental issue is that of course antibiotics aren't perfect.
Right.
And in the case of C. difficile, there's a rising incidence of vancomycin resistance. Some research reports on that anyway.
Well, this is the standard of care. The standard of care would be go with antibiotics.
Yeah, there's
More antibiotics for the problem that I got with my antibiotics.
Well, this is what's so insidious about the
Sure.
disease, right?
Yeah.
Because then you get treated and then you have recurrences. And this is the
Yeah.
big economic loss driver.
Because the vancomycin's also killing the things that were
Exactly.
creating the opening in the first
Yeah.
place. Right.
So the 3 antibiotics most commonly used are vancomycin, fidaxomicin, and metronidazole.
Okay.
And in the biggest, most tightly controlled study was run by the big pharma company, the antibiotics primary cure rate was only 80%.
Okay.
But it's okay. It's not the end of the world. You can switch to other antibiotics or load up the things, but
Or try again, maybe.
yeah. But still, that's a remarkable fact. I mean, antibiotics, we're accustomed to just kind of working right off the bat, but it's not true in all people, and particularly immunocompromised people like the elderly. So what I'm winding up to here is we're hoping in our trial to also help that rate of primary cure as well as preventing the recurrences. And the goal, and this is why we spend so much time making the cocktail as potent as possible.
Or the sort of other products that are working, or the other candidates that are working through the market, are mostly, they're going to come after, well, I get my second antibiotics, then I'm going to try to colonize, and hopefully the good bacteria will grow back faster than the bad bacteria will grow back. Whereas you want to hammer down the bad bacteria the whole time.
Yep.
And maybe this is synergistic with also implanting some good bacteria, but
Exactly.
while like at the same time as you're suppressing the bad bacteria, seems like it should work a lot better.
So in our dream world, of course, what we want is to, of course, our drug is going to solve all of the problems in the world, but we don't need that to be the case.
Because your drug is half of the golden duo that is yours plus someone else.
So in our trial, we're dosing it concomitantly with antibiotics until people finish their course.
Mm-hmm.
And the goal there is to accelerate the antibiotics. Of course, we can get people off the antibiotics, which have their own toxicities and side effects and things.
Mm-hmm.
And then we'll continue dosing for another 8 weeks in the main cohort. And that takes advantage of the fact that unlike antibiotics, which are very non-specific and they blitz everything.
And if you're taking them for 8 weeks, you'll mess up
if you keep on the antibiotics. So what we want to do is provide during that 8 weeks, you take advantage of that, the property I was talking about earlier, the specificity of these large macromolecule drugs, the 4 parts of our cocktail, to just keep out the bad bugs while the commensal bacteria are regrowing. So in our ideal world, then we will find, or we hope, that synergy with not only internal synergy with our product, but also synergy with the antibiotics. So we want to improve antibiotics outcome. And there's no incompatibility of our product with the FMT based approaches too. So altogether, our goal, I think, as an industry should be 0 CDI cases and 0 CDI recurrences.
Yeah.
And there's a lot of excellent ways to combine these things, in the future to maybe accomplish that.
So this seems like, in some ways, just a perfect combination of a bunch of things going right. This is an infection in the gut. Somehow the spirulina goes straight to the small intestine, which is where I wanted it to be. You've got these great antibodies. You've got 3 sites on the toxin that you can make use of. Do you have other programs running? Can you give us a taste of what those are like and the breadth of the approaches that are enabled by this manufacturing breakthrough?
Yeah, so it's pretty broad at this point. The common feature is it's essentially any disease that has some kind of a nexus. I'll
Okay.
just put it that way with the GI tract. So starting from the obvious ones, so C. diff we spent a lot of time talking about. There are other infections of the GI tract.
Sure.
They're all amenable to the same approach. The very obvious one, anybody with kids has had the pleasure of enjoying norovirus infection. Yep, it's like COVID. It's a virus, an RNA virus, not a bacterium.
And then someone's just going to have
But also
GI events for a couple of days.
if you think about the GI lumen as being our kind of natural home, then you can also think about the respiratory lumen, which would be the upper respiratory tract.
Another part that's outside of the body, but
Yeah.
enclosed.
So there, maybe an example would be RSV or influenza, right? Influenza is a virus that goes in there, so we can put the neutralizing antibodies into your nose.
We've had some recent experience with the third virus that ends up in your nose.
So that's a very interesting one because SARS-CoV-2, COVID-19 comes with a tropism. They call it. Where does the virus attack you? Is the tropism? The tropism for both the nasal passages and the GI tract, actually.
Yep.
Yeah. So that's a twofer.
Sure.
And we have a program running in that area with DoD funding.
Where you've got some spray for the nose and some pill.
Only we could formulate it also for the GI. So yeah, and then there's also the other external surface would obviously be the skin. And so we have a DoD funded program that's preclinical still, but that's also looking at antibiotic infections of like in battlefield medicine. There's a big concern there.
And these are similar approaches. You've got an enzyme that attacks
It's an enzyme or an antibody, sometimes a mix of things.
Where's that enzyme coming from? You've got some AI system that did something, or
Well, it depends on the program. We try to mine Mother Nature as much as possible because it's the only thing with more compute than
AI labs? Yes.
Yeah. So there's a lot of, in many of these cases, for example, the C. difficile program, the enzyme there is derived from something called a bacteriophage. Bacteriophage are the viruses, but the viruses they can't do anything to humans. They only attack the bacterium that they are co-evolved with.
The viruses of C. difficile that don't know how to interact with human cells.
So this is an enzyme that virus normally makes for its own reasons, but it turns out to be, if we just manufacture it recombinantly in spirulina, feed it to people, it pops the cells very efficiently. Yeah, kills them off.
And you've got a mix of similar tricks for battlefield infections for wounds.
Similar, yeah.
Sure.
Yeah. But it's all, now you can kind of feel the rhythm of what we're doing here, because in battlefield medicine, it's the same thing. There really are variants there. And so we need to cover those variants, but now it's with a cocktail of enzymes, not a cocktail of antibodies. Okay, so then one step further than that would be, so these are all like infections of the GI tract. Or traveler's diarrhea, that's another one. That was funded by the Gates Foundation originally.
For the benefit of the travelers, or is this just the name?
It's a nuisance for travelers, but it's actually the Gates Foundation's interest in this area comes because there's an incredible amount of infant mortality in the developing world. Caused by the
things that are nuisance for first world adults. The things that are nuisance for first world adults do kill kids in the third world.
So that's their interest. The same basic approach. Antibodies. We need to identify On these bacterial and viral pathogens, you can add norovirus and rotavirus to this there. A cocktail of antibodies that will bind and neutralize all of their virulent factors. It's a term of art.
Sure.
So these are all what's called non-host directed. These are things directed at the pathogens. And what they all have in common up until now is that these are all like basically swimming around in the GI tract or what have you. But there's no reason with a protein you can't hit some target that is related to your natural biology. So a very obvious place to go from C. difficile infection, which is great because we have all of this data that neutralizing this TCB protein with an antibody can help people. So it makes it much easier and lower risk for us to develop things. Well, there's another disease that's very common in the US called inflammatory bowel disease. It's quite similar. It's an inflammatory disease, not an infectious disease. But similarly to C. diff, there's an enormous amount of clinical data telling us what kinds of things to neutralize, or say things to neutralize. In that case, it's not toxin B of C. diff,
but it's like body systems that are going out of whack, and you need to.
It's immune protein signaling cells called cytokines.
Okay,
the obvious ones
but they're made by the body.
They're made by the
No
body.
problem.
Yeah, so IBD is an out of control immune reaction that happens in your GI tract when you outside of a context of an infection where you want the immune response. This is where it's gone a little bit haywire. There's lots of autoimmune diseases. Rheumatoid arthritis is a common one. So this is a very
the complications of COVID are the escalation of the immune system mostly.
Yep. Yeah. Exactly. Yeah. So here what we're trying to do is turn down the dial on the immune system that's causing it to go haywire and overreact when there's no illness, no infection happening. And so there, the cytokines neutralize the very obvious ones. Without giving too much away, we haven't launched this program into the clinic yet, but the very obvious ones are it's a cytokine called TNF, tumor necrosis factor, and another one called interleukin 23. So again, takes advantage of some of the same principles here. We know Humira, for example, is a TNF neutralizing antibody, the best-selling drug in the world up until Keytruda.
Yep.
So we know that helps IBD patients. And IL-23 is also marketed under the name Stelara. And in fact, we even know because Janssen ran a combinatorial trial, just 2 antibodies, not with the artful combination like we did, but 2 antibodies. And we know that together they get an even better response than any one of them, either of them alone. So that gives us a lot of optimism for that program. There are other programs like that. If you step back again, zoom out one more level, you say, well, okay, so we can maybe address these targets not only within the GI lumen, but also in the GI tissues. And so what other targets are there in GI tissues? And it turns out the GI tract is a pretty interesting interface of signaling and perception for all kinds of bodily systems. So, for example, the immune system, if you talk to immunologists, a lot of times they'll actually just come out and say it. It's like, oh, well, that obviously makes sense, Brian, because the GI tract is the largest immune organ in the body. So many subsystems within the immune system are interwoven.
There are
tissues.
There are immune systems, there are white blood cells, there's whatever happening as part of the line of defense.
Exactly.
Okay.
Constantly monitoring what's going on in the GI tract.
Right.
So
this is the defensive line of the human for the football.
It's also a sensory line.
Okay.
Yeah.
Sure.
So very far afield from anything we're doing, there's a line of research that's going on right now into the importance of commensal bacteria, the microbiome for all kinds of things. And there are some papers that, for example, have found interesting differences between the microbiome composition of people who respond to checkpoint immunotherapy and those that don't.
This cancer treatment.
Yeah.
Okay.
So it's totally different because you wouldn't say, well, that cancer is not GI cancer.
It's not in the GI, it's in your somewhere.
But nevertheless, you see there's something interesting going on. And the conjecture is that, well, this is because it's the largest immune organ. People are thinking, well, gee, if we can modify the microbiome in such a way that we can cause more people to be responders to checkpoint therapy, then that would be huge. We're not an area we're working in ourselves, but just I'm showing this as an example of the unexplored biology that is now unlocked by the Lumen technology, because now we can actually maybe start to, with precision, interrogate these things. Until now, you had your choice was either, nuke the whole microbiome and hope for the best, or intervene in pretty clumsy ways. Most probiotics really are just, just to be honest, they're almost entirely lactobacillus.
Okay.
Now, lactobacillus.
There's like one bacteria, but it's the most common.
It's the easy one to make into a probiotic.
I see,
So
right.
it's a yogurt.
Yeah, great.
So this can't be right. We would have known by now if it was a miracle cure for everything.
Right.
Lactobacillus is probably more complicated. There's trillions of bacteria down there.
Yeah.
So that's one example. There's the immune system, the nervous system,
Okay, go on.
or the vagal afferent nerves are all tied up in bound up in the GI tissues. Again, sensing what's going on, reporting to your brain in ways that we don't really understand what's going on down there.
But the idea that that's pressing on neurological disorders or
Yes,
propensities there?
that's
All right.
what people have proposed, and even some chronic diseases are proposed to have gut-brain axis origins. Parkinson's disease, for example, is one
Right.
that you see written about. Again, we're not doing any work in this area,
Sure. But it's exciting that this is elsewhere in gut biology,
Right.
like
Yeah.
happening.
And then there's the endocrine system. Obviously, the endocrine system is also all bound up, and this
Right.
actually makes very direct sense, right? Because
Well, yeah.
nutrient sensation
Yep.
and satiety.
But there's been some recent interest in the field of manipulating the gut for satiety and
A little bit. A lot of people don't know this, but the famous GLP-1 drugs,
Mhm.
the GLP-1 in your body comes from intestinal L cells,
Right.
the cells lining the GI tract.
Yep.
They're the ones that source the.
And someone has a molecule that just interacts with it in the right way, and it seems like a story that we've heard before on this episode.
Yeah.
Yeah.
So yeah, all of
So you've got work going on there?
we do have a preclinical program going in the weight loss space.
Okay.
I can't say too much about it, but it's very encouraging.
Sure.
So all of these are just kind of examples of the things we can do. But there's one more example which we didn't start working on ourselves directly. It was actually a Japanese pharmaceutical company approached us with this idea. It's a pretty good idea. Diet has an impact on a lot of things in our lives.
Mhm.
And a surprising one is kidney stones. So again, kidney stones are not a GI disease.
Right?
It's pretty far from the GI tissues.
Sure.
I haven't heard anybody describe the kidney gut axis, the gut brain axis, as a lot of followers. And nevertheless, about 90% of the kidney stones are comprised of something called calcium oxalate. These are crystals, actually.
Sure.
And in most patients, there's an orphan indication called primary hyperoxaluria, where there's a genetic disorder that interferes with the body's ability to break down oxalate. Oxalate accumulates in the blood serum, then accumulates in the kidneys, and then becomes supersaturated, mixes with calcium, it creates these crystals, actually. And then it's
I don't want crystals in my kidney.
one of those breaks loose. Yeah,
Yeah.
it's incredibly painful. Well, but actually, most of them don't come from this genetic abnormality. There's other things going on, and they have gut maladaptation, gut malabsorption syndrome.
That is causing this buildup of things.
And people, they absorb too much oxalate from the diet, so it's even harder to avoid than gluten for celiac disease patients. So, one simple approach this Japanese pharma company approached us with, they said was, well, why don't you just make some enzymes that degrade oxalate before it can be absorbed
in the gut.
in the gut? Yes,
Right.
just cut it off at the pass. As it were.
Yeah.
That's another great example. I think, in short, everybody we talk to has new ideas, and a lot of them are really, not all of them are good, but a lot of them are really good.
Yeah.
And the sense I get is that this must be what it felt like to be at Amgen in the early '80s.
When you can do engineering for the first time, you can
Yeah.
manufacture biologics for the first time.
It's not like all of those things were great ideas, but look, some of them were amazing. Enbrel, the patent on Enbrel was filed in 1989, and it's still a bestseller.
Yeah.
So we're very excited about the scope of the opportunity, but also the tremendous unmet medical need that is left lingering out there, despite the fact that the newest of our three big, reigning champion drug development technology platforms, the injection biologics, the newest of them is 50 years old now.
Right.
And there's still a lot of human disease.
Yeah.
And it seems like, wow, we've got this new tool that allows us to go after some of these old hard problems in a totally new way. I think we're going to find like Amgen did in the 80s. There's some really great applications
Right.
somewhere in there.
Yeah. So this feels tremendously exciting. You've described, by my count, 6 different drug programs at different stages of things. Most biotech startups, before they've gotten their first drug to market, they've raised a monster Series A round so they can get one drug through its next stage. Then they raise a monster Series B round and get it through. They raise a monster Series C round. It's just dilute to get just one drug passing through. So what is happening here that you're running so many programs in parallel? What's making that possible? Is it just the economics of how cheap any one of them has gotten? You can be doing 6 at the same time more leanly than anyone else.
It's part of that. So a lot of the cost comes from complexity.
Okay.
And I saw a newspaper article in the trade press a couple of weeks ago. I don't remember the name of the company, but the title just blew my mind. The headline was, such and such company announces $200 million seed round. Right?
What?
Yeah, it's incredible. It's a seed round.
Wow. Okay.
Million.
Okay. Why did they need $200 million to before they filed for their first in human?
These funds have gotten so big.
Right.
So back when I was practicing law, a typical biotech fund was $300, $400, $500 million, like the big ones.
Right.
And then they would do financing, equity financing deals that would be 5 million, 10 million Series A.
Sure.
Now that's not even a seed round.
Right. No.
So yeah,
Yeah, 5 is maybe a pre-seed just to get something done. But by the time you're at A, 30, 50 is what people say.
It's incredible.
Right.
Yeah. So we've never had that kind of money. Not for a lack of trying.
Sure.
But we can talk about that later. It's not really a fit for what the VCs look for and want to do.
Well, it doesn't look like everything else.
It doesn't look like everything else.
And when you have $500 million and you're deploying it 50 million at a time or whatever the heck it is, they're mostly looking for things that look like the last big success.
Yeah. Most biotechs, my sense is most of the time there's an idea, maybe like a hit non-optimized drug candidate from a university maybe. And then it's almost always something that uses an existing technology. So maybe it's a monoclonal antibody that there's lots of suppliers in the ecosystem that can work with that and manufacture it for you and things. And so things they all look kind of the same to my outsider eyes.
Yeah.
Not to say that other people aren't doing interesting things. I mean, there's an enormous amount of diversity. I think particular like Arch and Flagship and Atlas, Bruce Booth's outfit in Boston. I think they're doing tremendously innovative things. You've got,
and we have new modalities breaking through. People are using mRNA for the first time, and
vaccines. You've got the cell therapies and the gene therapies.
Yeah.
So all of that's true, and yet if you look and you kind of count noses, almost all of the companies are small molecules or monoclonal antibodies.
Sure.
Almost no vaccine companies aside from COVID era. So we never had.
Most of them are raising 10 million, 100 million
exactly.
at a time. Yeah.
And so we never had that kind of funding, and so we couldn't execute that kind of business plan.
Right. What do you do instead?
Well, so where we started instead is we had the core technology. We had the cell engineering technology, and rather than saying here's our drug for this disease, we need some money to go do the trials, we said, well, here's a type of cell we can engineer. What might that be useful for?
Right.
And it's a hard question put that way
to
answer, because it actually is useful for many things.
Right. But where do you start?
And human disease, by the way, is only one of the categories. And there's all kinds of industrial biotechnology, there's vet pharma, there's foods and food additives, you name it. There's all kinds of things, and all of them come down to biomanufacturing using a massively scalable microbe like we've got. So it is useful for all of those things. We can't do all of those things. We had to figure out what's the killer app? What would be where should we start?
Or it's just a beachhead. It's similar to
best thing to do, like Genentech did. What's insulin? But here. And it was our good fortune that we're here in Seattle, and some folks from the Gates Foundation wandered across, and they told us all about these poor kids in the developing world dying of these infant diarrheal diseases, which used to be a driver of high infant mortality in the US too. It's just that we figured out other ways of solving that problem. And that's what kind of cued us in on the opportunities within the GI tract, which otherwise is a bit of a afterthought in big pharma. There are no like GI health companies with $200 million seed rounds. That's just not a thing.
Yeah.
So we're kind of rotated 90 degrees in a way that isn't appealing to VCs. And so we said to the folks from the Gates, like what we say to everybody that has a great idea, got any money? And they said,
They have some money.
They do. And so they actually funded some of the first development work on this, and it was our good fortune. A lot of sweat and tears goes into this too, but things worked really well right off the bat. It's a great idea, and that's what sort of set us down on this path. But we started out on this path though, and we never. The Gates Foundation doesn't write $200 million seed round checks.
No.
It's
They'll get you one stage,
small money,
one stage down.
Real money where I come from, as the saying goes, but not this kind of thing. And so we've always had to be parsimonious with our spending. It just happens to be our good fortune that is what our technology platform excels at.
Economy.
Yeah.
Efficiency.
Efficiency. Going back to the kind of the origin story, which is we've got this way to make oral protein drugs, but we want it to be a couple of orders of magnitude cheaper than a conventional sterile fermentation system. Well, in order to accomplish, you can get 10% of the cost out of a system by just continuous improvement. And they've been doing that. I mean, it's much cheaper now to work with those systems. But if you want to take 2 orders of magnitude out, you can't just do the same thing in a more efficient way.
Right.
You have to wildly rethink things. And in particular, you have to think about things with a relentless focus on simplicity and robustness. And so of necessity, that is where our organizational DNA has evolved. Now, if you
as a company, not just in your science, not just in your
exactly, and it's everything. It started on the kind of the manufacturing side of it, but now it's how we design clinical trials. It's how we think about developing preclinical data packages. It's how we do the protein engineering. I mean, you name it. Everything is focused on high throughput and low cost. And I can't speak for other companies, but my sense is that when I was at a very well-funded company before this, and when you've got that much money, it changes how you approach problem solving. And a lot of problems you try to solve with money. And the thing is, most
you spend your money on outsourcing. This is, talk to other companies. A lot of the complexity comes in with like, well, we have all this money. We'll outsource this job. We'll outsource this trial, the manufacturing. But this is not how you're going about things.
Well, if you think about it, just doing something yourself versus sending it to a supplier, you can look up what the typical gross margins are of these companies. It's about 50%.
Yeah.
And that tells you, if you're on the math backwards, that it's twice the price.
Right.
You hope you get in exchange for that premium pricing, better quality, better speed, et cetera.
Well, that's if you're doing exactly what they were already ready to do. If you're doing something different, you will get charged something even worse.
So the insight though is even more fundamental. It's just that if you have a lot of money in the bank, it ends up being the lever you pull first, pretty much always. And almost all the problems in life can't be solved with money. It's just true in business as it is in sort of family life.
Yeah. Yep.
Most things aren't money problems.
Yeah.
So we had the blessing and curse of never being tempted with that.
Sure.
But it's also the technology itself just sort of naturally lends itself to this way of thinking. Now we try to go out of our way to optimize for it in kind of a more intentional way.
Sure.
And we try to root it in. But that is best I can do as an explanation as to how it is. We have so many programs and still a pretty small team. We're just a little under 100 people. But with that 100 people, we're doing actually many more activities than many much larger companies, which much more outsourcing do. So, for example,
in-house manufacturing
we have in-house manufacturing. We have our own GMP manufacturing facility that we own and operate exclusively for ourselves. We actually have two GMP facilities now.
There's the one it's a converted bakery here in Seattle.
Yeah, we built another one, Kitty Corner, in the 1441 building.
Got it.
Sure. Commissioning that now. But we also do all of our own hit to lead discovery, protein engineering, cell line engineering, all of our own regulatory work we do in-house directly with our own people. really the only segment of the, sort of a fully integrated biopharmaceutical company we don't have is sales and marketing. 'Cause you don't have a first
You don't have a
yet.
product yet.
Yeah. Yeah. But now when we kind of ease into a new area, this is the lens we bring to our eye, which is
Yeah.
okay, this feels like we could spend any amount of money on this.
Yeah.
But how are we going to do it for 2 or 3 log cheaper
Right.
than is the standard thing? And it always comes down to the same thing, which is without compromising, patient safety or anything like that, or scientific rigor, right? So we don't cut corners there. But what are the inessential bits? And there's a lot of inessential bits.
Mhm.
And it's really difficult, in short, to discover what those are if you're not forced to by, capital constraints.
Of all the things that you're running in house, clinical trial operations, I mean, are you contracted out to CROs there, or are you in direct contact with PIs at sites?
It's a hybrid.
Okay.
So for our late stage trial, working with an excellent group called MedPace. So they're based in the Midwest. I think that's maybe not a coincidence. They have kind of good Midwestern values.
Sure.
And
So you come to them with
Seattle has a similar cultural vibe
you come to them with this idea of efficiency, economy.
and rigor, because that's
Parsimony.
late stage trial, and particularly they have a lot of uh, recent experience running very similar types of trials in C. diff, and the direct relationships with a lot of the investigators at hospitals across the country where this trial is happening. So that's an example where we're really heavily leaning on the ecosystem.
Sure.
But for our earlier stage trials, we tend to be much more directly involved. We run them almost exclusively in Australia,
Mhm.
which is a bit of a distance and a complexity, but it's just a much more efficient regulatory environment for spinning up early stage trials and small trials. And what we really like there's a second aspect of what we like about Australia is it's very easy to have a kind of a direct relationship with the site PIs. And that's really important, we think, for particularly early drug development, because if there's one predominant story about great drugs, it is that how many of them come out of serendipitous observations.
Mhm.
Right?
Yeah.
Viagra and
Yep.
Rogaine are two
Yep.
famous examples, but there's many more.
But if it was just by the numbers, like, number of
Mm.
endpoints, number of adverse events, you'd see a lot of adverse events and walk away from it.
Yeah. So the approach, particularly in the US, where there's so much money available, it kind of complexifies things in a way where it's difficult to actually see what's going on
Mhm.
directly. And it's a real shame, because I suspect we're missing out on some of these kind of opportunities for serendipitous observation.
Yeah.
When everything is stripped down and standardized and stamped out, and sent out in the standard fashion to the same CROs that run all of the same studies, and they all run in the same way. I mean, nobody's really got enough of the pieces in front of their eyes to see the big picture like this, which is what you need if you're going to get those serendipitous observations.
And this is a question that I'm sure we'll ask a bunch on the show, but, what is the original sin of why clinical trials cost so much and take so long, and we're spending, how many tens of billions of dollars and getting how many drugs out the end? But it sounds like your answer here is everyone just has too much money.
It could, I mean, it's a bit perverse to say that,
Sure,
right?
yes.
As you know, like right now, there's this terrible capital drought happening in biotech.
Sure, yes. A couple of years now, it's
Yeah, and there's a lot of companies failing. I think last I looked up the numbers, it was something like a quarter or a third of Nasdaq biotech companies have market caps lower than the amount of
and the
cash
amount of cash.
they have in the bank.
Yeah.
Which is a brutal number.
Yep.
So yeah, it's a problem. I think even more fundamentally, you're probably familiar with this. It's kind of a catchy term. This researcher Jack Scannell came up with, called Eroom's law.
Yep.
It's Moore's law spelled backwards.
Yep.
And it's an observation that, unlike in microchips, which get cheaper and cheaper exponentially, clinical trials and drug development costs seem to get more and more expensive.
Yep.
Exponentially. And then Deloitte, the big accounting firm, they run an annual study, and they go crunch the numbers from public biotech, biopharma companies. And what they found is that there's a pretty persistent secular trend, maybe 20, 25 years now, of declining profitability. And they're looking at not only the cost of development, but revenue side. And that's because the cost to develop one new drug has been going up, and the market size achievable with each new drug on average has been going down.
Mm.
And that's not a sustainable pattern.
Sure.
Yeah. So, yeah, there's a lot of discourse about what are the ultimate drivers for it. The one you said, which I think might be part of it, is not one of the typically cited.
It's not commonly
explanations.
thrown around that everyone just
Yeah.
yeah.
But I like your thinking there. I mean, let's imagine let's imagine that's true.
Mhm.
What we would imagine happening? Well, all of those trends hit some kind of asymptote when COVID happened, because not only did everything become more expensive with
Right.
inflation, but particularly the inputs to biomedical research, even pipette tips. Remember, there was
Yes.
the great pipette tips crisis of
Yep.
2021.
And when vaccines were coming out, is we can't make enough syringes.
The syringes and the glass, and it's always something. So yeah, there were shortages of everything, and then the prices skyrocketed,
Right.
along with the price of lumber and other stuff.
Sure.
So now we're very much at the other end. There's a glut of lab space.
There's too many pipette tips.
There's too many pipette tips. There's like, basically, if you go through and you think about like Econ 101, what are the factors of production?
Mhm.
And all the layoffs mean that there's tremendous amount of human capital
Mhm.
around. All of the company shutdowns mean there's a surplus of lab space in Seattle, something like over 40% unoccupied lab space rate in the US,
Mhm.
In Seattle, rather, which is crazy.
Yeah.
So, yeah, what are the other inputs to product capital? Capital has gotten more expensive, but that's Jerome Powell's fault.
Sure.
But yeah, basically everything else has gotten cheaper. And I think it'll be interesting to see, and it's tempting to draw an analogy to something that happened in another industry sector that had a similar thing happen to it, where the cost of developing a .com
Mhm.
in the late 90s went through the roof.
Yeah. Yeah.
At the time, of course, you had to most of that venture capital money with these huge rounds,
Right. Right.
and then immediate IPOs, and well, what was all that money going to? It was going into the hands of Cisco Systems,
Mhm.
yeah, right, and Microsoft
Yep.
to
Yep.
buy the server chips and the software and thing. So it was all going into this picks and shovels
Yeah.
kinds of places. And then what happened? Right, 2001, it took a while,
Mhm.
a brutal adjustment. But what happened is then it was an extinction level event for not only the .coms, but also the VCs that invested in those .com. Most of them didn't survive.
Right.
They're like gone, forgotten now. But what happened is afterward, you had the mammals that were scurrying around, and one of the mammals was Amazon Web Services, wasn't it?
Yeah.
Right? And that set the stage, because now all of a sudden, people didn't have the capital to buy all the server racks from from Microsoft and Cisco and
IBM.
And instead, if you wanted to start an Airbnb or Netflix, you had no choice.
Right.
And importantly, I think you didn't have the investor community barking down people's throats to buy Herman Miller Aeron chairs,
Mhm.
spend the money, because remember this whole thing?
Yeah.
Spend the money, it's a gold rush, and not spending the money fast enough.
You're not hiring enough people. Well, just hire more people.
Yeah. And all of a sudden it became capital efficiency and minimum viable product, and this whole new way of thinking about it.
Mhm.
And there's this whole blossoming. In fact, all of the web companies that are dominant today, the Amazon is a kind of a partial exception.
Mhm.
But even them, without AWS, what would Amazon be today? Google, Airbnb, Uber, then the rise of mobile later. But so maybe you're onto something.
Yeah.
And maybe then the capital drought will, in our world, have the same impact in the long run. And boy, wouldn't that be nice? Because the potential for human flourishing is much
Yes.
greater in
Yes.
biopharma than on streaming media and taxi rides.
Maybe less facetiously, there's Bern Hobart has a book, Boom, about investment and innovation and any number of things. But he puts forward the hypothesis that, you really see flourishing of new economies, of new sort of realms of activity when you have this period of overinvestment.
Mm.
You build all of the stuff,
Yeah.
then it collapses.
Yeah.
And then people have to build up within this ecosystem where there's just lab space everywhere. But people have to be lean.
Yeah.
There is something appealing about that, well, we have built all these labs. Maybe something good will happen from all these little mammals scurrying around.
I hope so.
We'll see.
I suspect it's not going to be the $200 million seed round company.
Some people still can do this. They raised $1 billion fund, and like, I don't know. The top of this market seems not to have stopped. It'll just keep going. But the middle is having a different time than they were in 2021. I want to end on a few questions about the future, looking
Okay.
forward to sort of past this. And wild speculation, allowed, encouraged, and authorized. If your ability to manufacture cheaply at scale scales up to solving enormous tranches of the health problems of humanity, what is the scale of that manufacturing enterprise? What does growing that much medical grade spirulina or whichever organism it is look like? What's your constraint turn into? Is the constraint like raw materials, carbon, nitrogen, and oxygen? Is it the doubling time of biology? Is it the photons that you need to pump in for the photosynthesis?
It's the photons.
It's the photons.
It's the photons. Yeah.
So are you going to need, you're like, if this keeps growing, you're going to serve the world's metabolic disorder needs and their GI needs, do a bunch of stuff for cancer, for respiratory disease. You're expanding and you need facility after facility. You're going to need a nuclear reactor or three, as the data center folks are learning. Yeah. Do you have a vision of the world where this becomes the third pillar? Where this sort of scaled agricultural manufacturing, or yeah, what is different about that world?
There's something interesting about our industry. It's easy to be impressed with these big pharma companies. I mean, some of them do really impressive things. I mean, really, Novo done with the GLP-1 drugs.
Yep.
Pretty remarkable. And yet, how many trillion-dollar biopharma companies are there? Zero. Yeah.
Yeah.
And the stability among the incumbents is really pretty profound too. Like who are the $100 billion newcomers?
Moderna.
Nope, no longer.
Well,
Amgen, and Regeneron.
Yep.
60 billion. Close. We'll round up.
Fine.
So, and there's another. So I'm going to tie this together. There's another. I'm doing the weave.
Yeah.
There's another interesting thing, which is who buys Amgen's products versus who buys Microsoft. Microsoft is a $1 trillion company.
Mm-hmm.
So who buys? Everybody uses Microsoft products,
right.
And it's not just Americans, but it's Americans predominantly who buy Amgen's products. Mm-hmm. And so, adding it all up, where are we going? Why can't we be the first $1 trillion pharma company? Why not? If we're right, and this is as impactful as mammalian cell engineering and the first wave of biotechnology, I think there's easily enough unmet medical need to support that kind of value creation. But we're not going to be able to do that unless we're actually making products that serve the entire world. But the good news here, so this is where it comes back to your point, is I think we have a unique opportunity. Our products don't cost as much to make as Amgen's, which means we can sell them at a price that the global middle class can afford.
Right.
And it means they have to be great products because budgets are much tighter over there than in the US. But that's the scale that we think on.
Where the global middle class is often an afterthought in medical development. Where like, hey, yeah, we'll get to them after we're finished getting our money back from the Americans first.
Yeah. But the middle class in China is as big as the middle class in the US. It's like a lower, sort of purchasing power.
And 3 times as large.
Yeah. So okay, so that's where we want to go. Now, how do we service all that? Well, it'll be a network of manufacturing sites. Like, just look at what Lilly's doing now. They're putting 26 billion.
Mm-hmm.
But the good news is that, remember, our capex is a small fraction of what theirs is. So it's definitely doable. And in the medium term, we can service all of that here from Washington State. To answer your question, the main input, the main constraint is the photons. And the photons we get by conversion of electrons.
Mm-hmm.
And the electrons we get from these hydroelectric plants in the Columbia River that were overbuilt, essentially, in the 30s and 40s and have a lot of surplus power.
Sure.
So those would be great. I grew up here in Washington State, so this would be great for our state as well. Those would be great jobs.
So we'll be turning the energy of the Columbia River into medicines for at least a couple of doublings.
And then for the future, people post-COVID especially, we really want to have regionalized manufacturing. So our interest is, of course, we don't want to have our photons coming from coal power plants. That's not a good look.
Yep.
And anyway, solar's cheaper.
Yep.
So where do they have cheap power? That's not from an embarrassing source. from a, building a consumer brand here too.
Yeah.
Well, Iceland,
Mhm.
geothermal.
Yep.
Spain, it's
Mhm.
very nice.
Yep. For solar.
All right. The Middle East.
Yep.
Has a tremendous
Amount of solar, which is not normally where we think of the energy is coming from.
Yeah. Well, a lot of the energy comes from there, but solar is increasingly important over
Yes.
there. So yeah, so these are the kinds of places that we're looking to expand in the future. I mean, it's a little bit cart before the horse because we don't have our first approval yet, but yeah, that's very straightforward from a capital expenditures perspective.
On the other side, on sort of the consumers that we're reaching, if we live in a world where therapeutic proteins, whatever proteins you want, cost $10 a gram, what is the single thing you are most excited about that being possible? For
Well, our
human flourishing, for health, for you and your kids.
our costs at scale are already well below $10 per gram equivalent pure protein, and at those levels, then here's an idea that gets. I find it very discouraging to think about the reimbursement system, how drugs are purchased and distributed and allocated. Then there's a real policy choice in the allocation
Mhm.
made by insurance company, formulary managers,
In
also
cookie back rooms away from
exactly, and also by government agencies, also without a ton of scrutiny,
Mhm.
and not just in the U.S., also in Europe and other
Mhm.
places. So I like thinking about the opportunities for some of our products, particular so weight loss product is a great example because it's already hybrid market in the US.
that is going direct to consumer as well as through the old system.
If we up until now, it's been so expensive to make biologic drugs that the only people can afford them are ones with rich governments.
Mhm.
And it'd be nice if we can get the cost out of it to sell those at a larger scale, still do a good job for our shareholders
Sure.
who took a lot of risk,
Yep.
right, backing us. So we need to repay them, but so still have a viable business model, but sell it at a price that is affordable, independent of a government solvency, and thereby unlock a much larger
Sort of
market.
route around this system,
Yeah.
which is
So
sort of grown.
that's an exciting vision for the future. I don't think many companies are working on it, but I think people are heading in this direction right now. You may have heard about this. Lilly Direct, Eli Lilly they're selling their GLP-1 drugs
Yeah.
directly over the internet
For like 30%
Uh-huh.
less or something.
for 30% less. Yeah, and Novo is now gotten into the game too.
Sure.
Even more of a discount.
There's some arms race, and maybe they'll get to 50% of the price eventually. And
And so maybe they're leading the way.
sure.
Yeah.
Brian Finrow, thank you very much.
Well, thanks for having me. This was really great, Ross.
We've brought together an incredible group of people who are leaders in their fields. Each are coming at this from a different perspective, but the thing that unifies them is that they believe that something can be done differently, and they're doing it.
We think we can cut this Gordian knot,
To do it smarter and better.
Our equipment is even cheaper to grow.
Then brewing beer.
Yeah.
Excellent.
Yeah.
giving biotechs the ability to think and act more like tech companies.
These conversations are laying out the pieces of the puzzle.
The market is willing to pay for some of these premium vaccines.
Turn down the dial on the immune system that's causing it to go haywire.
We should be looking back and saying chemotherapy was barbaric.
We have an opportunity to unlock a better, healthier world.
This is not a zero-sum game.
This is Development and Research, an interview series about fixing the broken world of clinical trials.